Can everybody see? Or did I not share my screen? I think I did. Okay. So hi, my name is Yeji Lee, and I'm from Smith College. I am a junior studying biochemistry and engineering. And I will first present on behalf of Yerim Lee, who is from Carnegie Mellon University. She is a sophomore studying engineering. And yeah, I will be presenting on behalf of her because she could not make to the meeting. And she mainly worked on the types of vaccines that are being developed to combat SARS-CoV-2. And later, Samantha and I will present some additional information regarding these types of vaccines, as well as other group members. And so Yerim looked at an article from Nature Biology, right here. And it's titled "The Race for Coronavirus Vaccines." And it's a graphical guide by Dr. Callaway and his colleagues. And so I'll start reading her first note up here. And so virus vaccines use virus itself or in a weakened or inactive form. And so, sorry, a virus is conventionally weakened for a vaccine by being passed through animal or human cells until it picks up mutations that make it less able to cause disease. And Codogenics in Farmingdale, New York, is working with the Serum Institute of India, a vaccine manufacturer in Pune, to weaken SARS-CoV-2 by altering its genetic code so that viral proteins are produced less efficiently. And for inactivated virus, in these vaccines, the virus is rendered uninfectious using chemicals such as formaldehyde or heat. Making them, however, requires starting with large quantities of infectious virus. So you see the virus entering, making this cell, and then putting that into a human to trigger immune response. And so next we have protein-based vaccines for COVID-19. And protein-based vaccines inject coronavirus proteins directly into the body to stimulate an immune response. Alternatively, fragments of proteins and protein shells that mimic the coronavirus's outer coat is also being tested. So protein subunits, 28 teams are working on vaccines with viral protein subunits. Most are focusing on the virus's spike protein, or a key part of it called the receptor binding domain. Similar vaccines against the SARS virus protected monkeys against infection but haven't been tested in people. To work, these vaccines might require adjuvants, immune stimulating molecules delivered alongside the vaccine, as well as multiple doses. So this is a diagram. You see the spike protein added to the cells to be injected. And then the virus particles, empty virus shells, mimic the coronavirus structure, but aren't infectious because they lack genetic material. 5 teams are working on virus-like particles, VLP vaccines, which can trigger a strong immune response, but can be difficult to manufacture. So there's the diagram. And next, we have the nucleic acid vaccines for COVID-19. And these vaccines use genetic material, such as DNA and RNA, for a virus protein that induces an immune response. The nucleic acid is inserted into human cells, which create copies of virus protein. For many COVID-19 vaccines in development, the nucleic acid, codes for the virus's spike protein, both DNA and RNA vaccines. And these trigger CD8 plus cytotoxic T cell response to eradicate the virus, in addition to antibody and CD4 plus T cell responses. And this information was actually found in the article that Samantha and I read, which Samantha will introduce later. And so this is just a diagram. And this information is also from the article that Samantha and I read later, which Samantha will talk about later. And so DNA vaccines have higher stability than mRNA vaccines. The non-integrating feature of mRNA presents insertional mutagenesis, and the mRNA half-life, stability, and immunogenicity can be modified. And these are nucleic acid vaccine candidates in clinical trial that Yerim listed from Heart article. And so these four are the major ones. I can go and read it. So the mRNA 1273 COVID vaccine by Moderna, TX, uses lipid nanoparticle dispersion that contains mRNA encoding the stabilized perfusion SARS-CoV-2 spike protein, and it's in phase 2. And I think, actually, it should be in phase 3 because it's August now. And then this is another vaccine by BioNTech, Fosun Pharma, and Pfizer. And they're collaborating to develop a vaccine using lipid nanoparticle formulates, nucleoside modified mRNA vaccines that encode trimerized SARS-CoV-2 spike glycoprotein, RBD. And it's currently in phase 1-2 and testing 3 variations of the vaccines. And the preliminary report on BNT162b1 available for phase 1 and 2. And this is another vaccine that a lot of articles, actually, different articles talked about. And also Sai actually worked on this. And so the NIO for 4800 targets the Mars coronavirus spike protein, and the nucleic acid vaccine by attaching to the receptor binding site and neutralizing the epitopes. The vaccine has initiated a humoral and T cell response, as well as a spike in antibodies, which function via competitive inhibition of the ACE2 receptor, which the novel coronavirus also binds to. And this name is just another name for the same vaccine candidate. And it's also optimized through electroporation. And I wasn't sure what that meant, so I just included a parent note that discusses what that is. So, oh gosh, that's okay. Well, yeah, here's the information about that you could check out later, but it's from Wikipedia. And lastly, this vaccine by SimVivo was mentioned in the article that Samantha and I read, but I thought I would just mention it now since we're talking about the nucleic acid vaccines. And so this vaccine is a bacterial spike oral DNA vaccine encoding S protein of SARS-CoV-2, and it's under phase 1 trial. And so next we can go to virus vaccines. And wait, actually we did that, sorry. The order flipped. So we could go to viral vector vaccines. And viral vector vaccines genetically engineer another virus, referred to as vectors, to carry genetic materials from coronavirus and produce coronavirus proteins in our body. And these vectors are weakened and does not cause disease. So 2 main types are the replicating and non-replicating. And so for replicating viral vector, these newly approved Ebola vaccine is an example of a viral vector vaccine that replicates within cells. Such vaccines tend to be safe and provoke a strong immune response. Existing immunity to the vector could blunt the vaccine's effectiveness, however. And for non-replicating viral vector, no licensed vaccines use this method, but have a long history in gene therapy. Booster shots can be needed to induce long-lasting immunity. US-based drug giant Johnson & Johnson is working on this approach. Well, and these are examples of the viral vector vaccines under clinical trial. And so this was also done through a collaboration with different articles. So I'll just share yours first. So this vaccine by the University of Oxford consists of replication deficient simian adenovirus vector, named that, containing the full-length structure surface glycoprotein spike protein of SARS-CoV-2 with a tissue plasminogen activator leader sequence. And this vaccine expresses, actually this part expresses a codon optimized coding sequence for the spike protein. And this is under phase 3, I believe, by now. And it also has low seroprevalence. And, yep. And so next we have the 85 vector COVID-19 vaccine by CanSino Biologics. And this contains replication defective 85 vectors expressing the full-length spike gene based on Wuhan WHO-1. Phase 2 trials show vaccine dosage at 5 times 10 to the 10th power viral particles is safe and effective in inducing significant immune responses after a single immunization. And it completed phase 2 trials and it's planning on phase 3, I think. And these are some, So these are some of more information on this vaccine that other presenters will talk about. And we could move on to 8026 vectored COVID-19 vaccine by Johnson & Johnson. And this vaccine uses adenovirus serotype 26 as a vector, encoding a perfusion-stabilized SARS-CoV-2 spike immunogen, a protein which mediates entry of the SARS-CoV-2 into host cells. And a single immunization induced a neutralizing antibody response and complete protection against SARS-CoV-2 in 5 out of 6 rhesus macaques. I am so sorry if I'm pronouncing that wrong. This is not my note. I'm just presenting for our team member. And so this is under phase 3, I think, or actually it's starting phase 3 in September if results are positive. And lastly, this is another vaccine that the article that Samantha and I read about. And the 2 vaccine candidates are non-replicating biovector vaccine. And so for approach 1, they modified dendritic cells expressing SARS-CoV-2 minigens. And it's in phase 1-2. And for the second approach, they used artificial antigen presenting cells expressing SARS-CoV-2 minigens, and it's in phase 1. I think that's all I think I covered all the different types of vaccines. So yeah, that's it for me for now. Thank you.
All right, I can present next. I think I'll be talking about the development and challenges that come with developing a vaccine, manufacturing problems, protective immunity, and a couple other concepts. So. All right, just to clarify, you guys can hear me, right? Someone just type something in the chat.
Yeah, we can hear you. And also see your screen.
Awesome. So yeah, awesome work with describing the different kinds of vaccines and the different ways they can be administered. So I made a quick list, a relationship describing the different vaccine candidates with a couple front runners, namely the one from Moderna, University of Oxford, Novavax, and Novio right here. And then I made another relation node to describe the development of COVID-19 vaccines. And this lists the challenges, antigen selection, protective immunity, which I'll go into further a bit later, and the preclinical evaluation. This basically describes different studies to actually describe if the vaccines are truly safe or not, and manufacturing. So let's start with that. I actually wanted to start with the clinical trials over here. And to go a little more in depth, a phase 1 trial is more or less based on the different dosage administered based on a vaccine. So, and they're not random samples. So they're a selected sample, and the different dosage is applied for the drug. just to see if there's any positive effect and to test a humoral response. Phase 2, the evaluation is expanded to more people, and that also includes a different separation by age, sex, health, and status of their health as well. And then finally, like a bunch of frontrunner vaccines currently in the market are not in the market yet. A lot of the frontrunner vaccines that hopefully will be in the market. Phase 3 vaccines are health trials are thousands of candidates that are given the vaccine, and then these results and the data accumulated from all of these trials are then sent to the World Health Organization to be judged and then tested, and then sent to the FDA for their testing and to see if it's safe. So to go back here, I'm going to start with the challenges of vaccine development, namely antibody-dependent enhancement. So this is considered probably the biggest obstacle right now. This is basically when antibodies of the viruses bind to the host, and then what happens is the host cell then will just open up a molecular pathway, allowing for more of the virus to enter the cell, and then this can allow lysis or a lysogenic cycle, where the virus just continues around its generation cycle and produces more viruses. And this has been seen with yellow fever, Zika virus, the novel coronavirus, and the ADE effect has to be like constantly monitored and see how that affects patients, because this could cause an inflammatory response rather than a humoral response, which would be beneficial to a patient. So that's the main thing about challenges. I also talked about challenge studies. Challenge studies are basically studies that are trying to prove why vaccine selection might not work out. This is also like you could characterize this under like a random sample study. So this would be affecting patients of all forms equally. And then comorbidities would, here, let me see if I have a separate note for that. Do not. Oh, awesome, Sam. I guess the cold chain for vaccine seems to be a separate example. I'll let Sam present that if she wants to. But then, yeah, I'll go on to the next section, vaccine antigen selection and design. Basically, this is selecting what components of a virus can be used in a vaccine. And yeah, and this right now the most common one is using the spike protein, because that binds to the ACE2 inhibitor or receptor and host cells. But along with that, if I'm not wrong, the E protein, the M protein, and protein coding for the nucleocapsid, which encases the virus, are also being targeted. And new ones also include the B and T epitopes of the virus. So those would be considered different binding sites, which could be used for vaccine. And yeah, another factor would be protective immunity. So this is basically what a successful vaccine should have. I think I have another note talking about what a successful vaccine is. No? Okay. But again, it should elicit a long-lasting immunity as well as a humoral response. And this should be through a release of T cells, but at the same time not overexpressed, so you don't have an inflammatory response. And at the same time, you also you want to avoid a cytokine storm, which has been covered in like I think every focus group in one place or another. Actually, I will try to see if there's something in physiology which I could bring up real quick. And this one pretty much summarizes it really well. Um, Patients with cytokine storm syndrome have a hyperactive immune system, so an inflammatory response, and they release a high level of cytokines and other immunological mediators, and this causes harm to the patient. Um, out of here. Ah, here, navigating is a little trouble of mine. But yeah, that covers the development of COVID-19 vaccines. I do plan to keep adding to that, but that's basically the kind of outline for it. So you have the description of the description of the clinical trials, what they are. Oh, this is the successful vaccine. So what I was trying to say earlier, that was linked to the whole preclinical evaluations, that safety of the vaccines, as well as protective immunity, is that currently under Section 564 of the U.S. Federal Food, Drug, and Cosmetic Act, the commissioner of the FDA can remove some of the legal obstacles and regulatory obstacles for a drug to pass if it's on the grounds of a public health emergency. And what, of course, that seems a little vague, and I don't know exactly the requirements for a public health emergency, but this is a section which could allow a lot of vaccines to pass in the market, or the most successful one to pass within a record time. But the problem is the long-term effects of a successful vaccine, or a vaccine that elicits a long-lasting immunity response, isn't known, and it can't be studied, especially with the number of cases increasing on a daily basis, as well as the number of deaths increasing on a daily basis. But then the flip side, if you look at it, every day a vaccine is delayed, billions of dollars of economic damage. Well, of course, aside from that, damage to infrastructure and just people's lives are caused. And not only that, but this also brings into the question of patents. Now, if the person, or I guess the organization that produces a vaccine, produces a patent, there are several ways that they can go about this, and they could also not patent and try to make it just free access to try and get the vaccine to the market as soon as possible. But if there is a patent, that can also go two ways, where they have their name on the patent, which where it would also delay the process by several months. And if not, they could also just have, rather than having their name, they could also just have, well, basically it's a legal obstacle that could delay the release of vaccine. And that's another repercussion of a successful vaccine. And this all falls under the umbrella of the resources needed to produce a vaccine, and that's more or less the manufacturing aspect. So that is tying back to the whole immunity response and how long that's going to last, and how fast and effectively a vaccine can be produced while minimizing side effects and any other comorbidities down the line. Other than that, Yeji covered a lot of the vaccines. I think I think every vaccine in this list was covered under some concept / relation node. And the articles I summarized were under the TPC plan, but let me see if I can find it fast real quick. Yeah. So I summarized the promise and challenges in the development of COVID-19 vaccines, the evaluation of immunogenicity of prime boost vaccination. So this was, I think, for the Inovio pharmaceutical vaccine, as well as another article here, which is basically for the resources needed in the manufacturing costs or problems associated with producing a vaccine. And challenge studies here. Those were the main articles I used to produce the notes here. And that's my quick little presentation. Yeah. Anyone else want to present? It doesn't also have to be a presentation. If you guys produce any notes, you guys can just talk about that. We also can just discuss this week's topic regarding vaccinations and any questions associated with that. I know there's a lot of like, it's a very wordy topic, especially considering the molecular pathways involved. But yeah, feel free to reach out, ask any questions, say hi in the chat. Otherwise, we can move on.
Sai, I also have some other things that I could also talk about today if no one else wants to go.
Yeah.
So I can. Oh, Samantha, you want to present?
Let's go.
Yeah, I thought we were going to present.
Let's go.
I was like, ready.
Go present.
Okay.
Yeah, I thought you were going to present.
Yeah, well, are you starting or do you want me to start?
You can start with your notes, and then I just want to talk more about nanomaterials specifically. So yeah, do whatever makes you feel comfortable. I think.
Okay, awesome.
Talking.
Huh? Yeah. I said,
Great.
yeah, I'm going to share my screen. Yeah.
Okay, good. Thanks.
Okay. Hopefully everyone can see my screen. So just to like introduce myself again, I guess. My name's Samantha. I go to Syracuse University. I'm a rising sophomore, and I study biology and medical anthropology. So Yuji and I summarized this paper. One second, I'm just looking for, okay. So it was called COVID-19 Vaccine Development and a Potential Nanomaterial Path Forward. So just to start off, so about the development of the vaccine, so for challenges of development as well as an aspect of manufacturing, Sai just briefly brought this up, but just to like reiterate, cold chain is, it refers to the maintenance of consistent temperatures for refrigerator vaccines from manufacture through delivery to healthcare facilities. So this can be an issue with poorer countries in the maintenance and distribution of the vaccine, because even if they develop a vaccine, being able to bring it to these countries and to bring it to different places and a lot of different populations and still maintain the cold temperature can be a potential obstacle. And then in terms of manufacturing, some features for the production platforms. So the production platforms can include like mammalian cells, yeast, bacterial cells, insect cells, and molecular farming in plants. And we just listed the advantages and disadvantages from a figure in our paper. And then to get into the types of vaccines, so we broke this down into two types. They're live attenuated vaccines, or LAVs, which are live reproducing. And by mild vaccines, that just means that they won't likely give you the disease if you get this vaccine. So they're meant to produce single dose immunity without illness. So that would obviously be the advantage, is that you won't get sick from taking the vaccine. The most prominent one right now is Codagenics Incorporation's proprietary de-optimized SARS-CoV-2 vaccine. And then some disadvantages of LAVs can include cold chain, which we went over, risk of viral transmission, risk of reversion to pathogenic form, reactivation in immunocompromised individuals, recombination with related viruses circulating in the population. So that's for LAV. And then the other type would be your inactivated vaccines, or your IVs. And these are pathogens or fractions of pathogens that have been inactivated by heat or chemical reaction. So some advantages to this, well, one of them that we found is that it's more stable than LAV. And some disadvantages is that it can have a lowered immunogenicity. It can be a requirement, so there's a requirement for a multiple dose regimen to establish long-lasting immunity. You may require adjuvants to immunize the aging population, and again, cold chain is another thing to consider. And then some that are currently in clinical trial for SARS-CoV-2, we just listed them here. And you can see they're each made by different institutions, and they're mostly in phase 1 or phase 2. And then this last one is just in phase 1 of development. So that's for the vaccines. Now we can talk about the viral vectors, the viral vector vaccines. So this is from Yaram. She just listed them here. But then the advantage to one of them, which is 85 vectored COVID-19 vaccine, the advantages would include broad tissue tropism, adjuvant qualities, and scalability. And some disadvantages would, well, one of them that we found is that there might be pre-existing immunity in humans, which might dampen the effects of the adenoviral vector. And then finally, just to go back to the S protein, or the spike protein for vaccine development. So in SARS or MERS-CoV-2, the spike proteins are responsible for the host receptor 28 binding and priming by host proteases to trigger membrane fusion. And so this protein is an attractive target for vaccine design because it allows the virus to enter the host cell during infection. And so I talked about this a bit. So the majority of COVID-19 vaccine candidates target the spike protein and receptor binding domain of the virus. And there are two methods for processing and presenting antigens to the human body. So nanocarriers are needed to be degraded for encapsulated antigens to trigger the cellular immune response, while the surface displayed antigens primarily lead to humoral immune response. And then some current studies identifying immune epitopes are the Lajola Institute for Immunology, the University of Cambridge, and Immunonomic Therapeutics, or EpiVax, or PharmaJet. So that's what I have from our paper. I can hand it over to Yeji if she wants to cover the rest of it.
Thanks, Samantha. Yeah, so I'll be talking more about nanomaterials and how that could be used to modulate immune response once they're administered to our body through vaccines as adjuvants, as Samantha mentioned. So I will share my screen again. And so nanomaterials have emerged as promising tools to either amplify or suppress our immune response. And by acting as adjuvants added to vaccines, engineered nanomaterials can aid subjects with impaired immune systems, such as the elderly, and to reduce the amount of vaccine protein production and injection. And similarly, researchers have developed lipid and lipid-based nanoparticles as delivery platforms for mRNAs and siRNAs to enable the synthesis of key viral proteins for vaccination or to inactivate critical viral target genes, respectively. And so there are ongoing screenings and classifications of various nanomaterials based on their immunomodulatory properties and cytotoxicity levels. And developing an adjuvant for clinical use requires phase 3 randomized trials, which could, take several years. And also, I was also working on this node with another member in our group, and her name is Noel from Smith College. And so I worked on the first half, and she worked on the rest of it. So I just wanted to give her credit. And for examples of nanoparticles that can modulate immune responses, we have these ASs, and these are licensed adjuvants that have already been found to stimulate early STAT1/IRF1 interference signaling, and I'll talk about that later. And alum is another example, and this is the most commonly used adjuvant in human vaccines, and it triggers the release of cytokine in macrophages, and for specifically the expression of IL-1 beta, which depends on NLRP3 inflammasome sensor signaling. And saponin-based matrix M is currently currently in phase 1 clinical testing as an adjuvant for the glycoprotein nanoparticle vaccine for COVID-19. And these are more examples. And so this one was interesting. Graphene oxide functionalized with amino groups, and this nanomaterial can boost immune response by activating the same signaling pathway that I just mentioned up here. And so in monocytes and T cells, and this results in accumulation of T cell chemoattractants and polarization of macrophage 1-T helper 1, and this is another nanoparticle that the article that Noel and I were reading. And this functionalized nanodiamond promotes anti-inflammatory response in mice by reducing macrophage infiltration of lung capillaries that may lead to difficulties in gas exchange. And so the article that we were reading is called, sorry, I just need to look for it. I forget where I put the citation, but it's called Toward Nanotechnology-Enabled Approaches Against the COVID-19 Pandemic by Dr. Weiss and his colleagues. And I hope, well, okay, there you go. So yeah, that's my citation, and that's it. I just thought that it was interesting how they were using nanomaterials to sort of play with your immune response. And that's it. Thank you. Does anybody else have anything to share with us? Or if not, we can end our recording here.
Wait, you didn't want to discuss about, hold up, good question.
Oh yeah.
Yeah. Like, is everyone staying or planning on continuing when school starts?
Oh, I think we should end the recording before we talk about it.
Wait, like the interns included though. Oh,
Huh?
yeah, I forgot that. I forgot that.